Good morning, everyone. I'm Danya Jose. I cordially invite you all to the international webinar on Super Resolution Microscopy organized by the PG Department of Physics and Research Center, St. Xavier's College for Women, Aluva. Kindly cooperate with us and follow the guidelines for the proper conduction of webinar. During the session, keep your audio muted and video switched off. Don't present your screen during the webinar to avoid unnecessary disturbances. Kindly write your name and address in the chat box after the recording gets started. This would be an hour-long talk followed by an interactive session. Participants can post their queries in the chat box, which will be discussed at the end of the presentation. Interaction with the speaker during the presentation will not be entertained. Towards the end of the webinar, the feedback form will be shared in the chat box and WhatsApp group as well. You can fill the feedback form for getting your e-certificate. Now, I invite Dr. Sheena Xavier, Head of the Department of Physics, St. Xavier's College for Women, Aluva, for the welcome speech. Sheena, ma'am.
Thank you, Danya. Good morning to all. Respected Principal, Dr. Sister Gigi Jonama Xavier, esteemed resource person, Dr. Annie Augustine Jose, Vice Principal, Dr. Sister Stella, and dear participants. Although this long stretch of lockdown has disrupted the normal academic process, it has to be seen positively, since the teaching and learning communities has accepted the medium of technology. This has also led to more opportunities to empower teachers and students by interactive webinar sessions with experts in the field. We have a wide spectrum of students and teachers attending this webinar. I take this opportunity to cordially welcome our dear principal, Dr. Gigi Jonama Xavier, who is the source of inspiration for all our endeavors. Hearty welcome to you, sister. Today we are quite fortunate to have Dr. Annie. Okay, sister. Today we are quite fortunate to have Dr. Annie Augustine Jose as our resource person to introduce us into the topic, Super Resolution Microscopy. Dr. Annie Jose is now a postdoctoral fellow in France after completing his PhD from Institut of Optics, University of Bordeaux, France. He completed his master's from International School of Photonics, QSAT. He has published several papers in conferences and journals. Dr. Annie Jose was introduced to us by Dr. Vimala George of our department. We are very fortunate, sir, to have a person like you amongst us, even for this short period of time. On behalf of St. Xavier's College and every participant of this webinar, I extend our warm welcome to you, sir. Dr. Sister Stella, our energetic and vibrant Vice Principal, has kindly consented to grace this webinar session by her presence and blessings. Hearty welcome to you, Sister Stella.
Thank you, Dr. Sheena.
Extending a hearty welcome to all. Thank you, Sister. Extending a hearty welcome to all the participants. Thank you all. Over to you, Danya.
Okay, ma'am. Thank you, Sheena, ma'am. We have with us our pillar of support and constant motivator for all our initiatives, our beloved principal, Reverend Sister Dr. Gigi Jonama Xavier. Let us hear a few words from Sister. Sister?
Yeah. Dear resource person, Dr. Annie Augustine, and dear participants. Let me first extend our greetings from St. Xavier's College for Women, Aluva. As we all realize, the spirit of lockdown has changed the whole pattern of approach to the process of teaching and learning. In this situation, where nobody can predict the return to the classical approach of face-to-face academic transactions, the positive transformation shown by scholastic community is the extended use of technology. I take this opportunity to congratulate the Department of Physics for organizing this international webinar. I am quite happy to welcome Dr. Annie from France, who will guide us in this webinar on super resolution microscopy. Dear Dr. Annie, we are really honored to have with us, and also we look forward to your assistance in future. Thank you all.
Thank you, sister, for your valuable words. With no delay, let us start our session. I hand over the platform to Dr. Annie Augustine, sir. Let us begin the session, sir.
Okay, so good morning, everyone. First of all, thanks to the PG Department of St. Xavier's College for organizing this event. It's really refreshing to see how the intellectual vigor is being kept even during these difficult times. So I will, without any ado, I will start the talk. First, I will introduce you to, I will talk about a problem that we have in science, about the scale of the natural world that we interact with. So as you can see from the scale of planets and usual other structures that we're familiar with, the niche that is occupied by humans it's very small. It's very specialized, and our senses are only tuned to operate in this small range of length. But there exists a different or completely exciting world beyond and above this scale of our scale of range. So, for example, if you look into the microscopy world, it is like in the range of micrometers and nanometers, which are like incredibly small compared to our experience of our normal life. And this small dot that you see represent the limit of what the current optical microscopes can see. But already it's very small, but you can see that some of the very interesting stuff like the viruses or the I'm sorry, I think you have a problem with viruses or the proteins that are very interesting for the study of in biological applications lie beyond this limit. So in order to study these systems, we need to develop new techniques to go beyond, and the scale extends far beyond this limit. So there is no ultimate technique that will help us to study the whole range, but we have to devise different techniques for different ranges. So I just wanted to give you a feeling of the scale of the experiments that we are dealing with, and how do we interact with this diverse field of environment. So, for example, there are no the only way or the most successful way that humans have devised so far is through the electromagnetic spectrum. So a lot of processes emit electromagnetic radiation, like there are sources for the radio waves, microwave and infrared and radiotherapy and high energy gamma rays. And utilizing this spectrum has been the most successful endeavor in the science that extended our knowledge beyond what was possible in our small scale of our existence. And also, even in this spectrum, only a very, very narrow range of wavelengths are accessible to us, which is the visible range. And beyond that, there's a huge swath of other wavelengths which are not known to not experienced by us, but are incredibly useful. Like for astronomy, people for centuries were using only the visible spectrum, but once we the X-ray or the radio wave astronomy became available, we got access to so much more knowledge. So what I want to emphasize here is that electromagnetic spectrum is the best way that we can or the most successful means that we are used to study the scales ranging from be it stars or down to the cells, because there is no sound waves or any other detectable thing that we can get from these different scales. So in the case of astronomy, if we go to the large scale, suppose we are observing something in the cosmos, like a faraway galaxy, like millions of light years away. What we do is we collect the light, which is, since we can assume that there's infinity, we collect the parallel rays and we focus it using a mirror or even a lens. But mirror-based systems are much more common these days, and we focus it to a point and observe it. And in the case of a telescope, since the objects of interest, the things that we are observing, like stars or be planets or galaxies or black holes or whatever, they're so far so that we get only a fraction of the light that is traveling, that's coming to us from there. So the race to make better telescopes have always been based on how large mirror that you can make. So you can see the evolution of the mirror size, like from 1893 to the proposed new extremely large telescope of 2025. The size is what is increasing. So the more the size of the primary mirror, which is in this case, this is a primary mirror, the more light it can collect and more signal that you can, more faint structures that you can observe. So in astronomy, always the problem has been how much more light you can collect. Resolution was also a problem, but mostly the collection efficiency. And even in these cases, some of them are land-based and some of them are space-based. So this is a James Webb telescope, which is due to launch next year, which is very exciting for all of us, which is space-based telescope. But in the case, this is in the extreme large end of spectrum, where we observe things that are in a huge incredible, in the scale of light years or even millions of light years. But when we go down to the scale of cells or atoms in the micron or nanometer range, or micro, where the name microscope comes from, what the microscope basically does is that it takes an object, which is whatever, something associated with a cell or bacteria or protein, and it magnifies the image, and you can take that image in a camera, or you can see using your eyes. But all the modern systems use a camera, and this system provides a magnification. But there is what is emitting the light. In the case of astronomy, the stars are emitting light, and we just have to collect it. We have to collect it efficiently. We just have to collect a lot of them. But in the case of cells or in the lower end of spectrum, there is nothing that is emitting the light. So in the beginning, people used to look for a different contrast enhancing agents, like absorption. Like if some parts of the cell absorbs more than the other, it can give a contrast, so you can see some parts better than the other. So it can work as a distinguishing feature. And people then went to polarization, where different parts of the cell can, or the tissue can impart different polarization, and looking into the differences, you can infer the difference. Or even phase, where the most successful was the phase contrast methods, where you actually use the phase of the light, which is shifted due to the presence of the organelles or tissues, and use that to impart contrast to it. But in general, compared to astronomy, the microscopy was lagging, like far behind, because the contrast was not as high. Because if you're looking at stars, you're looking at a bright object against a dark field, which is like the contrast, the ratio, a highest ratio. It's normalized to 1. But in this case of cells, if you are looking at phase or polarization, there is not enough contrast. Always the problem was contrast. So the problem was, how can we get a light source inside the cell? How can we put an agent that we want to observe inside the cell? And if you're observing a protein, how can we make them emit light? And in the 60s, there was a very interesting observation in the jellyfish known as Acera victoria, and they the researchers found that in this jellyfish there was a protein called what they named as green fluorescent protein, which can absorb light at around 480 nanometer and have emission of 520 nanometer, which is a phenomenon of fluorescence. So this is a naturally produced protein. The jellyfish can by itself generate this protein, and they can under the under the UV light they can see the fluorescence. And even though it was invented in the 60s, they had back then there was no good means for sequencing or just to do the fusion protein. So it was in the backwaters for a long time. But in the 90s, precisely in 92, people were able to fuse this protein, this GFP protein. So you can see the structure of the GFP protein in this figure. So it's a beta barrel protein, where inside the beta barrel there is a chromophore, and this yellow agent inside that is the fluorescent part of the protein. So when you excite it with a blue or UV blue violet range, this chromophore inside will emit at will absorb that radiation and fluoresce at closer to 520 nanometer. So in 1992, they were able to fuse the protein, to fuse the GFP protein to a protein of interest. So proteins are synthesis by reading a code and arranging the amino acids in the right order and folding. All the folding is a big part, but we'll just look at the sequencing part, the sequence part. So what they did was that they edited the sequence that is read to make the protein such that it can encode a GFP protein also in. So when the protein is expressed inside the cell, as for example, in this case, you can see the protein called thalin, which you can see in orange color. So this protein was the coding for this protein was modified such that when the protein is expressed, it will be expressed along with a small molecule of the GFP protein also. So by naturally, if you excite this this protein, it will emit the fluorescence. So we can see this protein against a background of the dark. So what we essentially made was we put an agent of our inside, and we can excite it with the blue region of light and collect the fluorescence. We look at only the emission of the GFP so that we don't see any other light. So essentially, we are having the highest contrast available. So a bright emission against a dark background. So this more or less solve the problem of contrast in imaging. So in the left, you can see it's a bright field image. It's based on absorption. So the some parts absorbs more can be seen as darker, and other parts are lighter. But in the right, it's the same cell but fluorescently labeled. You can see that if you label for a factin, you can and the nucleus, you can see them with much better contrast than the bright field counterpart. So the fluorescent label, the protein labeling, more or less solved the issue of contrast in microscopy, and it was comparable to other observations. And so once the problem of contrast was solved, then it was a race to the bottom. So because even the cells, they are very small. It's in the micron range, and the proteins inside them are even smaller in the range of nanometers. So how small can you image? And this is where they stumble into a fundamental limit called diffraction of light. So we'll just so the fluorescence. Once the fluorescent proteins were established, people were able to devise many different imaging schemes. So in this case, I will just show you how the people were able to establish confocal and wide field microscopy. So in a wide field microscope, you have excitation and it excites. So this white stuff is your the molecule or the protein which is fluorescent. So in white field, it excites all of them together and it emits the fluorescence, which can be collected by the camera, and you can see in the image, you can see the 3 molecules. But if you add a pinhole or 2 pinholes in the system, one for excitation path and one for the emission path, then you basically can make a point source, and you can focus this this beam to in sample, so that focus this beam to the sample as shown here. So only a part of that that fluorescent molecule is excited in this case. So only that part will be emitting. So you can see that when we collect the fluorescence, we can say that, oh shit, sorry. You can say that there is only one fluorescent molecule, and by scanning the beam or moving the sample, we can image the 3 of them. So this was like the introduction of confocal microscope really revolutionized the field of bioimaging. Until then, people were using like techniques from the 20s or 30s, but the introduction of confocal imaging brought about like a real that thing you can see and quantify. And also, since the beam is focused, it don't excite the molecules which are out of plane. So it gives sectioning to the images that you take. You see only in one plane, and you can move the sample up and down to get stacks of images. So sectioning and resolution are 2 important contributions that came about with the confocal microscope. So basically what we do is that we use a focused beam and we scan either the beam or the sample and record the fluorescence from each point using a camera or detector. But so suppose you have the stars are a fluorescent molecule. So you have the beam, the focus beam passing over this fluorescent molecule. So if they are like sufficiently spread apart, and the beam is passing over them, you can see 2 peaks corresponding to when the beam passes over the. So when the beam is over the first molecule, it will emit some signal, and you can detect it, and sequentially the second one. So you get 2 peaks, and you can when you reconstruct the image, when you scan the entire image, you can see 2 blobs like this. But once it comes closer, these peaks come closer, and the image also seems to be very close, and in this case is considered to be a limit of resolution. We can say that there is 2 of them, but if it's beyond that, if they are even closer than that, then it will appear to be as 1. And the limit is usually it's said when the maximum coincide with the first minimum. That is usually said to be the limit of resolution. And this limit was first introduced or described by Ernst Abbe, and this is a picture of his grave. It was such an important discovery that they engraved it on his grave. So the resolution or the limit of diffraction, the smallest distance that you can separate between the two emitters, are decided by the wavelength that you use for imaging. So if you want to do like cellular or live cell imaging, that if you want to image live stuff, this has to be in the visible or near infrared region. So you are limited in the range of 400 nanometer to maybe 800 nanometer or 900 nanometer, sometimes even more. So you're limited in lambda in that region, and n sin theta is the numerical aperture, the collection efficiency of the system. So this is you can play with the n, which is the refractive index, a little bit, but there is a limit. Even the best systems can have a n sin theta of 1.6 without introducing like very serious aberration. So in this equation, the lambda and the denominator and numerator are more or less fixed. There is a limit for them. So for a typical value of lambda that is used in bioimaging, the diffraction limit comes out to be about 250 nanometer. This is for the for the lateral direction, so 250 nanometer in the lateral direction, and it is a bit worse in the axial direction. It's higher, but this is quite frustrating because the diffraction limit is caused by the light. It's the actual protein that you're imaging is only 2.5 meters across and 2.5 nanometers across and 4 nanometers in length. So if you consider the diffraction limit and you put the protein in the center, it's like a tiny fraction of the PSF of the diffraction limit. So you are imaging something like this, but you are getting something the whole diffraction limited pattern. So that makes it very frustrating, because this limit is actually preventing you from seeing something that is actually very small, but it just spreads everything around a point spread function. And also in the third and lateral direction is close to 200 nanometer, but in the axial direction it's almost even much worse, almost more than twice worse than the lateral direction. And this is important for bioimaging because you can see there's one image of the lateral image and one axial image. So in the lateral image, you don't know what the object that you're seeing is. It might be a flat circle, or if we don't know the size of what is object is. So when you're doing biology, size matters. So you want to know what is the actual size of the object that you're imaging. So 3D the diffraction is messing up with our measurements in the three dimension much more than the lateral direction. So we need to have so people were quite frustrated. The biology and the imaging community was very frustrated by these limits imposed in the lateral and axial direction, and they've been looking for solutions for pretty long time. It's, from this the limits of the system, by the things that you can improve in the optics or in the detection or in the lenses, the quality of lenses, it was already reached in the 1900s. So for a long time, more than 100 years, I think, people have been trying to surpass the diffraction limit, but they've been stuck. So some of the techniques that evolved out of this struggle was, one of them was near field scanning optical microscopy. So in this case, they use a tip or aperture which is very small. So very close to this tip or this small aperture, the light is confined to a very small region, much smaller than the diffraction limit, which is related to the size of the aperture of the tip. So if you put your sample very close to this tip or this aperture, you can have much like higher resolution. In this case, they showed to be 50 nanometer, which is like 4 times better than the diffraction limit. It's already good, but the problem is this near field, the region where the light is confined is called near field regime, and this regime is confined to a very small distance from the tip or the aperture. So if you can just image the surface variation, the surface imaging only. If you want to go inside the tissue or inside the cell, it's not possible with it, because you can just measure only very close to the tip. And it's pretty, this technique is pretty difficult because you have to keep the tip in close proximity to the sample that you're imaging. And another technique that was developed for this super resolution was called structured illumination. This is a more interesting and it's more novel idea that was evolved. It was in the 2000, I think, they developed. And so in this scenario, in this paradigm of imaging, we look only in the frequency space of the image. So if you have a structure to image, maybe it's a cell, the SIM looks at the cell as a collection of spatial frequencies. And the closer the feature size, the two molecules are very close, then the frequency is very high. and if two molecules are further apart, the frequency is very low. And what the objective does is the objective has, let's say, low pass filter. It passes only the low frequencies, which means you can image only those molecules which are like very far apart because they have very low frequency. So all the high frequencies are cut off. So the problem, in SIM community, the problem was reframed as a issue of the objective due to, since it cannot pass the high frequency bands of the image. So how they overcome, or how they the technique that they introduced to surpass this the frequency, to increase the frequency bandwidth, was to structure the illumination. So when you illuminate the sample, instead of using a uniform illumination, you use a structured illumination. So your sample will have a periodicity, and the illumination will have a different periodicity, and those two will form beats. It will like the Moire pattern, and the Moire pattern will have one component which is of lower frequency. So actually you can smuggle this high frequency information inside the Moire pattern and take it to the objective, and take it to the camera, and using the post processing you can actually get this information back. So it was like the convolution and deconvolution thing. And this okay, so this effectively increased the bandwidth of the objective, but still Moire pattern, this frequency can only increase up to twice the actual resolution of the microscope which it already have. So there was a limit that you can go with SIM, but more modern SIM techniques like saturated illumination microscopy can go even further, but it's very difficult in its implementation, and people are almost giving up on this on the saturated structural illumination. But there are still new techniques going on, new reports coming from this field. But yeah, the problem was that we can go only up to twice the resolution of your microscope really are. And actually, for the pattern, you can actually use to do a lateral super resolution. You can make use a 2D SIM, and when you can use this, you can generate this pattern by interference of two beams. So you just focus two beams in the sample plane, and they will interfere, and this causes the stripes. And you do different stepping or different phases to collect all the spatial frequencies and collect it. Or if you add one more beam, you can have a structuring in the 3 dimension also. So you can have whole 3D super resolution like this. But SIM is actually not technically called a super resolution because it's still diffraction limited. It's still limited at twice the resolution you had. So it's still diffraction limited, but it's an extended resolution technique. So this is not, SIM is not considered true super resolution technique. As you can see here, if you image the actin filaments using the structural illumination, you can see, compared to the conventional wide field imaging, you can have much higher resolution in this case. You can actually solve the different filaments in this case. It was a neat technique, but yeah, it had its limitations. So in 2014, the Nobel Prize for Chemistry was awarded to 3 scientists for the contributions to super resolution microscopy. Actually, it's interesting because the 3 of them were physicists, but they were given the Chemistry Nobel Prize because the super resolution techniques that they developed were more a contribution of the probes, the new probes that were developed, than the optics. Because optics, the limits of the optics were reached 100 years ago, and the new probes were what enabled to do actual super resolution. So the super resolution is not diffraction limited. So the limits of the image resolution in these imaging techniques is not confined or not defined by the limits of diffraction, unlike the other technique. And one of the key features of all these techniques was to do super resolution, you need to need on-off transition. You need to have a probe or a protein which can be switched on and then switched off, maybe more times, or for some techniques just one time. So the key requirement is on-off transition, and these techniques were not diffraction limited. So let's first talk about localization of emitter. So if you have one protein which is fluorescent and you occur an image of it in the camera, then the precision of the localization precision can be defined by this equation, which can be approximated to this nil by the square root of the number of photons that you collect. So you can see that the localization precision increases, and the localization inaccuracy it decreases the more number of photons you collect. So the resolution, or how precisely you can determine the position of a protein, depends only on how much signal, how much photons you can collect from that molecule. So if you can have a very bright protein, then it means you can localize it much more, so you can have a much better estimation of its resolution. So the idea that they had for doing super resolution with this kind of probes, which this technique called photo activated localization microscopy, or PALM, was developed by Eric Betzig. So Betzig was working in Bell Laboratories on using its near field scanning optical microscope, but it was pretty frustrated by the development of the slow development in this field and the difficulty to do far field imaging. So he just quit his job one day, went his home, and then built like a new microscope in his living room, and you can see it here. So what they basically done is that they had a protein which was emitting in the green channel like GFP, but if you excite, or if you apply a violet radiation to it, it switches to the red channel. So if you have some structure which is diffraction, lot of proteins which are very close together, closer than the diffraction limit of the system, then you can actually convert them one by one. So you make sure that no two molecules are closer than the diffraction limit, and you find the position individually. Then you do the same procedure again, until you have matched all the molecules. So this way you can estimate the location of the molecule with a much higher resolution than what is allowed by the diffraction limit. And yeah, so what basically enabled us is that we were enabled this high resolution is that we were able to individually identify the molecules through the on-off conversion. So the on conversion was the photo conversion. You turn them to the red channel, and when you image, you bleach them. So all the fluorescent molecules will bleach after some time. It's inherent in all, unless it's very sturdy fluorescent materials like nanodiamonds. All the fluorescent proteins, they will bleach after some imaging. So bleaching was the off process, and photo conversion was the on process. And you find the location of each molecule individually by turning them on one by one and making sure that no two of them are closer than the diffraction limit. So yeah, this is the physics behind this is pretty simple. The setup, it's very, very simple, and they could just build it at their home. So the crux of the experiment came from the quality of the protein, the photoconverting protein. And after that, so you can see here that compared to the diffraction limited image, a PALM image can resolve the proteins much better. So here they use the double labeling of two different photoconverting proteins. In the green, you can see it's much better resolved than the other case, and you can see the colocalization of the other protein in a different channel. So yeah, you can PALM has been reported to have 20 to 30 nanometer solution in routinely in day-to-day experiments. So it's pretty good at resolving structures which are close together, like you see in this image. And soon after, many other techniques were also evolved. And one of the techniques that reported soon after was stochastic optical reconstruction microscopy, or STORM, where they use the blinking of the fluorophore. So the fluorophore can blink due to many reasons. And one of the reason they are during the cycling or the fluorescence, when they go to the excited and ground state, they can transfer to a dark state and stay there for some time. So they use special buffers to tailor the time that they spend in the dark state, such that when you look at the one molecule, you can see that it's blinking intermittently, and you can actually make sure that no two molecules are at the on state at the same time, at the closer than the diffraction limit. So if you can have a decent blinking efficiency, you can actually separate the molecules, just like in the case of the palm. You can separate them and individually localize them. So the technique was the same. You have on-off transition, which was photo conversion, or in this case, it's a dark state conversion, and you find the position of the molecule with the number of photons that you have collected. So and over this last 2 decades, there's been a lot of techniques, and they are all slight variants of the same core principle. Like you have an on-off transition. So we don't have time to discuss all of them, but they even have a technique called lobster. So the landscape of the techniques available is very huge these days, and there are a lot of probes available these days which are very sturdy, which can image for very long times. And yeah, but the basic idea always remains the same, which is you have a molecule, you turn them on one by one sequentially, and then you image. But still, we were just talking about only the 2D resolution in this case. So we were just fitting the number of photons collected in the image, so that we get only the 2D super resolution. But actually, the molecules could be stacked on top of each other. But in the case of palm or storm, there is no way to distinguish between if the molecule is in one plane or above that. Because the emission is symmetric with respect to the optical axis. So we need to introduce some asymmetry in the optical axis in order to distinguish what is the axial position of the emitter. So this one of the earlier techniques that was introduced was astigmatism. So you add a cylindrical lens which will induce astigmatism, aberration to the system, so that your PSF of imaging is not symmetrical in the axial direction. So in the plane of focus, it will be like a normal Gaussian. If you go above the plane or below the plane, they will have different structures. So from the ellipticity of this or elongation of this PSF, you can actually determine how far away it is from the sample plane. So this was combined with the palm or storm. The astigmatism can be used, and you can actually have a full 3D super resolution. And another technique for 3D distinguishing is supercritical angle fluorescence, or SAF microscopy, where you excite the cell like normally, and then you collect the emission, and you will use a very high NA objective, which is 1.49. So the objective will contain the supercritical angle of the emission also, and in the path you split it into two, and in one path you block the supercritical angle, and the other path you collect it. And if the molecule is further away from the cover slip, the contribution to the supercritical angle will decrease. And by actually looking at this ratio, you can find how much distance this emitter is away from the cover slip. So basically, this is just a technique to have a distinguish distinguish the distance of the emitter in the axial direction. And you combine it with the storm or palm, and you have all 3D super resolution. So this is a picture of a rugby game, which is very popular in the region that I'm living. I don't know the rules of this game, and if someone give me this picture and tell me, oh, can you find how this game is work, I mean, there's no way I can imagine. Or I will give you 10 images of very high resolution. Can you find the rules of the game? No, it's not possible. To at least to have an idea of what they're doing, we need like the dynamics. We need to see what the players are doing, which players are interacting with each other, and what they are doing in real time, to know to have a understanding of what are the rules of the game that is being played. It's the same in biology also. In the techniques that I discussed till now, the coordinate stochastic techniques, where you individually image each molecules, you lose the information about the dynamics. You are sequentially imaging each molecule one by one, so you don't know which components are interacting with each other, who they are interacting with, and what is happening in a dynamic sense. You just get a static picture of the final condition of the imaging, the subject of your image. So but to understand the processes behind the process that you want to study, you need to have the dynamics basically. So this is where different super resolution, different class of super resolution technique called coordinate target nanoscopy shines more. So in this case, it's a similar. What you see here is similar to a confocal setup. So you have a focused beam, and the beam you can see that it's focusing to a sample plane. And suppose you have two emitters, and when you scan, so you can see when the focus beam will have a PSF, elongated PSF with X, Y, and Z distribution. And when you scan the emitters through this beam, you can see it will record a fluorescence like this. So since these emitters are closer than the diffraction limit, you can see one big distribution. But suppose you have a secondary beam which can switch off the fluorescence, which can suppress the fluorescence, such that when the beam is present, there will not be any fluorescence. And also suppose that this beam features a minima in the center, like a hole in the center. So what will happen when we scan this together with the excitation beam? So in this case, when you scan the emitter over that same beam, you will see two peaks, because in the center, when there is no emitter, it was suppressed in both sides. So you can see two distinguished peaks when you scan along with the depletion beam or switch off beam. So even if the two emitters are closer than the diffraction limit, if you can have a suppressing beam, then you can actually suppressing beam with the intensity minima in the center, you can actually distinguish the two of there are two of them, not just one of them. So basically, this is also a on-off process. So you have an on process where it is the excitation beam, which is shown in the green, and you have off process, which is the red, which suppress the fluorescence. So the idea remains the same. You need an on-off process, and you can distinguish the two which are close to each other. So when you compare this technique to the coordinate stochastic technique like STORM or PALM that we discussed before. So if suppose you are imaging a dynamic scenario, like something that is moving very fast or reorganizing very fast, the coordinate target techniques, which since they are like sequentially imaging everything, they will give a closer to the accurate scenario. But in the coordinate stochastic like PALM and STORM, since you are sequentially imaging each molecules which are not spatially correlated, you mix up all the information. You superimpose the information from past, present, and future into one image. So you kind of blur out all the dynamics image. So PALM and STORM, they are like very ideally suited when you're imaging fixed cells which are not live. We fix the cell, you image them, they're not moving, you can localize them to very high position. But once you move on to like live cells and you are looking for dynamics of different structures, then you're at a loss, because there is no way this stochastic techniques can reproduce the information with sufficient clarity. So so now we need to have a off switch off process to turn off the fluorescence. And one of the most popular techniques in this coordinate targeted methods is stimulated emission depletion, or called STED. And in this case, the off process is depletion, stimulated emission depletion. So you excite your fluorescent protein, your fluorophore with the wavelength, and it will emit a fluorescence at this green, suppose, and you will be able to deplete the fluorescence by stimulated emission by using another beam of very high power, and with if you have an intensity minima for that beam, you can actually use it to image with a high resolution, like we seen in the movie before. So you just have excitation beam, you combine it with the depletion beam with the phase modulator, which imparts a phase such that the beam in the sample plane will have a intensity, or we call it donut beam in the real sense, that it has a zero, it has a minima in the center, and you combine the beams and it's focused in the sample plane, and you do the same as in the confocal laser scanning microscope, where you scan the image using moving the sample or moving the beam. And in this case, the resolution, it depends on the size of the donut, so how small the intensity minima of the donut beam is, and it is actually decided by this equation, where there is an extra factor of 1 plus I by I sat, where I sat is the saturation density of the molecule, and I is the depletion, the intensity of the depletion beam. So if you have like very high power available to you, like if you have a lot of intensity to give, then you can have high resolution. So this means that the resolution of your imaging is not defined by the diffraction. It's defined by how much energy you can put into your depletion beam. And normally, since you're depleting the fluorescence, and fluorescence is very short-lived, it's in the nanosecond, you have to deplete it very fast within that few nanoseconds. Otherwise, you will get the fluorescence. So you have to deplete it very fast. So you have to put very high intensity. And normally, usually, pulse lasers are used for this STED lasers, because they have very high peak power, and thus have such short, efficient depletion. And here you can see the nuclear pore complex image by using STED and confocal microscope. So in the confocal, you just see some blur. You don't know if there is any organization, but instead you can see there is a organization in the periphery, which where they form discrete structures, and in the middle where the second protein of it. This is a two color STED imaging, so you can see the localization of two different proteins at the same time, which is not at all available in the confocal regime. So STED has been extended to the 3D imaging also, which is was more or less that to produce a donut-shaped beam with a zero in the center, use a vortex phase mask. So it's a ramp of so phase mask is something that imparts phase to the beam. And if you have a helical phase mask, a ramp phase mask, then each point of the beam which is imparted by this phase mask will cancel out each other in the center, because each point which are opposite to one point is at a phase shift of pi, which is 180 degrees. So they will cancel out each other when they're focused. So in the center you have a minimum, and but in the axial direction there is no structuring, and you can add a structuring in the axial direction by using a zero pipe faceplate. In that case, you will have the interference will make sure that in the center of this beam there will be a minimum, and you will have two very high intense lobes in the up and bottom portion. So this is called a bottle beam, and this is called a donut beam, and this is your excitation. This is a Gaussian beam. So if you just want to do XY lateral imaging, you just have the excitation laser and the STED laser with the redshifted STED laser. And if you are doing 3D STED like XYZ imaging, you have excitation, you have a normal donut beam, and you have a bottle beam also. So the two combined will make sure that the fluorescence is suppressed in all three directions, and you have a close to isotropic resolution in all three dimensions. And also, it's only determined by how much intensity that you can put on your depletion beam. So for long time, nanoscope techniques started emerging in the 2000s. So it's relatively new. It's only 2 decades old. But in the earlier days, even until recently, people cared about only 3 aspects in the imaging, like how much contrast that you can have. You need to have the best fluorophore to have very high thickness, or have very high contrast, and how much temporal resolution you can have. Which means what is the fastest process that you can image with your system. Like if the dynamics of some structure is very fast, can you image them? Or the spatial resolution, how much resolution you can gain by using this technique. But more recently, a fourth factor was also considered, which is the specimen health. So when you're imaging, you don't know if you're changing any properties of the specimen. Especially in biological condition, when you put, like in the case of STED, when you put like insane amount of intensity for the depletion of the fluorescence, you don't know if you're killing or barbecuing your cell. So more recently, specimen health has also been considered to an important metric to decide how the quality of your imaging. So all the techniques that we have at the moment is a compromise between these 4 factors. So if you can have at least 3 of them very good, then that's good enough technique. And this is the compromise that you have to make when you are doing the nanoscopic imaging. You have to choose which one is more important for you. So we need to have a technique which can access the dynamic information. So that means it should be coordinate targeted. It should be using very low light intensities. It should have a high spatial resolution, and it should have high contrast. So we need to have on-off process. The on-off process for the fluorescent molecule that can be used to implement a coordinate stochastic method. So if you review the photoconvertible fluorescent proteins, you can see in the fluorescent molecules can be subject to various different kinds of photoconversion. One is photoactivation, where the molecule is off in the initial state, and it can be turned on by exciting it with a particular wavelength. Or photoconversion, which we see for the case of the palm, where it can switch the channel of its emission by using a particular wavelength. Or you can have a reversible photo switching, where it can go to the on state and come back to the off state and do it like multiple times. And you can have blinking, where the like we saw in the storm imaging, the molecule in the on state can go to the dark state, and you can come back and go back again. So these are the 4 major photo conversions in also photo bleaching, but we won't discuss it here. Out of this, the first 2 is routinely used for palm imaging, and the blinking is used for storm imaging. But the 3rd category is pretty interesting because it's a reversible on-off switching, so you can turn on and turn off. So you can use it for a coordinate target method. And one of the protein that is exhibiting this thing and this on-off behavior is a mutant of GFP called RSEGFP, which is a reversible, switchable, enhanced green fluorescent protein. And the photo switching is explained by a change in the isomer state of the chromophore inside the beta barrel. So the cis state is an off state, and trans state is on state in this case of this molecule. So using this, we can implement a imaging system called RESOLFT. RESOLFT stands for reversible saturable optical fluorescent transition. So it's like a the technique implementation is similar to the STED, but I use the photo switching instead of the depletion of the fluorescence. So what are the requirements for the prop that you use? So it should be having very high brightness, which means high quantum yield. So it will have a very high contrast and low bleaching. So it should not bleach very fast. You need to do the imaging for a long time and many cycles. And wavelength should be in the visible range. And there are biological requirements like it should mature fast and should have high stability and should be monomeric, otherwise you have other problems, and stable in the pH of your experiments, and experiment should be stable at 37 degrees where you do your live cell experiments normally. And in the terms of imaging, you need to have a lot of number of on-off cycles to do the super resolution. So it should have low switching fatigue, and the contrast between on and off state should be very high to distinguish between these two states. And the switching should be fast, and this should not take too long for the switching, and the off state should be thermally stable. It should not spontaneously come back to the on state during the imaging. So if you have a protein that matches all these criteria, we can implement a scheme where a protein that can be switched on using the UV wavelength or 4 or 5 nanometer, and it can be switched off using the 488 nanometer. So we use the same STED principle where the beam features intensity minima and UV readout. So switch on, switch off using the intensity minima beam, and read out using the normal diffraction limited Gaussian beam, and you will get the fluorescence only from the center of the beam. And you use it to scan the entire sample so that you can produce image. So when you compare the light dose that is required for different techniques, like the coordinate Sokais technique, the overall light dose is the light intensity that you need is very low because there is no depletion or switching off. But you have to do the imaging for a long time because you have to image all the molecules sequentially. So when you consider the overall light dosage, it will be very high. And instead, it's intrinsically very high intensity is required because you are switching off the fluorescence, and you have to do it very fast. You use pulse lasers, so the intensity use is in the thousands of kilowatts, even hundreds of thousands of kilowatts. But since you are doing it for only very short time, the overall light dosage is more or less similar to the case of palm storm. But in the case of RESOLVT, which uses the photo switching technique, photo switching requires very low intensity of light, only a few kilowatts for the switching. And you have the same pixel-wise imaging scheme as instead, so you don't expose the sample for a very long time. So the overall light dosage is like orders of magnitude, sometimes close to 6, sometimes order of magnitude less than other techniques. So when you consider the specimen health, RESOLVT is like a very reasonable technique because it puts only very low photo damage to the sample. The photo load that your cell or your tissue experience is very less because you're not putting too much intensity and not for a long time. So RESOLVT can provide sufficient high resolution, and it can have temporal dynamics because it's a coordinate target method. It can have a high contrast because using a bright fluorophore like close to the GFP protein, and the light dosage is low, so you can have a very high. The sample health is reasonably good. So using RESOLVT, we in our lab we've been doing imaging for quite some time, and you can see these are microtubules in a COS7 cell. In the diffraction limited regime, you see something like very blurry or very non-dissolvable, and you have a lot of background also. But when you do RESOLVT, you can see you can distinguish each filaments individually, and you can see the resolution is close to 42 nanometers, which is like almost 5 to 6 times better than the diffraction limit. And also you remove a lot of the background that you had because the emission volume in this scheme is much smaller, so you are getting the background only from a small region, which increases the contrast. Also, you can in the live cell you can see the dynamics of the filaments for a very long time. So this can be actually used to take long time lapses and see how the cells are behaving under different conditions. So until now, I was discussing only about a donut based resolved or STED setup. So you have only one intensity. There's only one zero. There's only one hole in the donut. But this need not be like that. You can parallelize it to whatever order you want. So this was part of my PhD thesis. We parallelized the resolved nanoscope using using a arrangement called optical lattice, where instead of just one zero, you can have hundreds of thousands of zeros. And this interference pattern, this optical lattice was created by interference of 4 beams. And you can see that the overlap of the two beam interference can be used as an optical lattice, which can which will feature an intensity minima and almost symmetric intensity distribution about the zero, so that you can have isotropic depletion, and you can use it for 2D imaging. So instead of looking at one point at a time, you look at a lot of points at a time together. So you can decrease the imaging time by a huge amount. So we create the optical lattice by interference of these 4 beams, and you can see this is an experimental image. You can see that it features the this intensity minima in a grid-like fashion, and you use a camera for the acquisition. So you put the interference lattice, you have a camera, and you acquire the images, and you put a mask, and just like a single donut case beam, you parallelize the thing. Instead of one donut, you use 1,000 donuts, which is created by the lattice, and you acquire it in a camera. So this can increase the speed of imaging by more than a factor of 10. And as we use a dual lattice to this is a technical aspect, but it was very important. We use the dual lattice for the activation. So you have to switch on the protein and then switch off the protein. So you have to use 2 lattices, one for switching on and one for switching off. So at the end, our setup had 2 lattices in a complementary fashion, which was so we could do very low phototoxic imaging for a very long time with very less bleaching and very high resolution. As you can see here, it's close to 40 nanometers, which is more than 5 times the diffraction limit. And you can see, we can look into the ultra fast dynamics of the vimentin filaments here. So the filaments that you see here, they are fluorescent protein, but they are endogenously labeled, which means it's relatively new technique called CRISPR-Cas9. So the cells by themselves are expressing this protein. The cells themselves are fluorescent. You don't have to modify the cells later. So this endogenous expression. So this is very close to the real biological conditions, and you can use you can see the live changes in the cell, and at the resolution 5 times better than the diffraction limit. So I will wrap up my talk by discussing the most recent and the exciting technique that's come out last year in the field of nanoscopy, which is called MinFlux. It's the most trendy technique that there is now. MinFlux stands for it stands for 2 things. It's not an abbreviation, but it stands for 2 things. It stands for maximum information and minimum flux. So the idea is that you get maximum information about the position of your emitter by using minimum photon fluxes. So how they do is that they use the same beam, like a donut beam, which has an intensity minima in the center, and they move the donut beam a little bit to the sides of the protein to see how the intensity changes. So if the emitter is in the periphery of the donut beam, you will have higher signal. If it's close to the zero, you have lower signal. And by using a kind of triangulation technique, you can find the actual position of the molecule by checking the signals of these four probing points. You have a donut beam, and you have a protein, and you probe the protein with your donut beam at different points, and different points will give you different levels of signal. And with this signal, you can actually locate the protein with very high precision. And the setup is very close to a STED or RESOLFT experiment, but you have a donut beam, but you have a like very advanced electronics and some XY diffraction systems which are not generally used in STED and SOLVE experiments, and you can have fast modulation and fast beam scanning. So using this min flux technique, people have actually shown resolution very close to, I mean, less than 1 nanometer in like calibration samples of DNA origami. So these are DNA origamis with the emitters attached. So you can see that using influx they can. So we already know that the emitters are placed like this in the folded DNA origami, and the influx actually produces the images just like that, even in this case. And you can see the fitting can give you full width at f maximum of no, it's a resolution of 0.8 nanometer. And even in live cells, which is more challenging to image than DNA origami because you have a lot of other problems coming associated with it, you can have a close to 2 nanometer. So the size of the GFP molecule is around 2 nanometer. So in this case, the min flux is pushing the limits of nanoscopy towards the size of the actual the emitter. So in the beginning, we discussed that the most frustrating part was that the size of the molecule was very small, but diffraction, the PSF was very large. So it was like messing up with all your imaging. But now we reach the limit of the molecule, the size of the molecule. So this could be the next barrier of imaging, which is right now nanoscopy has reached its limits. So yeah, with that, thank you for attention, and I'll take any questions that you may have in the chat.
Thank you, sir, for the wonderful session. It's time for the interactive session. Participants can post their questions in the chat box. Kindly post your questions in the chat box.
I think there are no questions from the participants. These are recent developments in the microscopy field, I think. And you have presented it very clearly.
I think we have a question here from Purnima Bishani. So
Yeah,
can we use and RESOLVT methods for live cell? Yeah, of course. People are using. I mean, I'm using RESOLVT for live cell imaging, and CALM technique they are using in live cell, not for structural imaging, but for diffusion studies to see the diffusion profiles of the different proteins. But yeah, they are widely used in live cell imaging. And the next question: at what temperature these experiments are conducted? All the live cell experiments are conducted at 37 degrees. That is the ideal temperature for physiological experiments.
So it seems the questions are over. Then
Okay.
thank you, sir, for your patience and willingness to clear the doubts. Now we have come to the end of the session. Okay, ma'am?
May I
Yes.
interfere? I have
Yes,
a different
ma'am.
question. I have a question different from your presentation. In your profile,
Okay.
I have seen that you have done a lot of summer programs in premier institutions in India, isn't it?
Yeah.
IISc,
Yeah.
BAC, etc.
Yeah.
I have seen the presence of a lot of PG students here. Can you advise and motivate the students to attend such programs? How you have got admission to those programs and how you have reached your present position? Please, sir.
Yeah, sure. So for the more intense, they have IAC have a very nice summer internship program where they take applicants and you have to write a small proposal. And I was selected through that program. And other ones like my master project or my PhD, you will find a field of research that you're interested in, and that then you read about the research and you get acquainted, and then you contact the professor and then talk with him. That's the best way. Once they know that you're interested in the work, they will also be interested and they will write back to you. That is my experience. But
Okay, thank you, sir.
You'll have to be very well acquainted with the work they are doing before you contact them. Yeah.
Okay, thank you, sir. Now we have come to the end of the session. I now welcome Dr. Sujata NB for the vote of thanks.
Okay, then. Respected Principal, Reverend Sister Dr. Gigi Jonama Xavier, our Vice Principal, Sister Dr. Stella, our dear resource person, Dr. Annie Augustine Jose, Head of the Department, Dr. Sheena Xavier, distinguished participants, and my dear colleagues. With constant enthusiasm, determination, hard work, and God's grace, the impossible has become possible. Ultra-high resolution observations of cellular objects is now made possible by the research in the area of super resolution microscopy. Today, we had a very good coverage of this novel topic. Dr. Annie explained us how this high resolution is made possible through the on-off transition and how it is used for the dynamic imaging. Switching over to my duty, I feel privileged to propose vote of thanks on this occasion on behalf of Department of Physics and on my personal behalf. First of all, I would like to thank our dear principal, Reverend Sister Dr. Gigi Jonama Xavier, for being with us with constant encouragement and support. Thank you, sister, for your gracious presence. Now, I would like to place on record our sincere love and gratitude to the resource person, Dr. Annie Augustine Jose, who is known for his passion for physics from childhood. In spite of being very busy with lab work after the 2 months of lockdown, he readily accepted our invitation and gave us a very good overview on the topic. On behalf of St. Xavier's College for Women, Aluva, Department of Physics, and all the participants, I extend our sincere thanks to Dr. Annie. Thank you, Annie. I wish all the best and God's blessings on all your future endeavors.
Thank you.
Next, I would like to thank our Vice Principal, Reverend Sister Dr. Stella, and our HOD, Dr. Sheena Xavier, for all the guidance, support, and motivations given to us. And now, the success of a seminar is its participants. Department of Physics, St. Xavier's College, express our sincere gratitude to all the respected participants for your active participation in the seminar. My hearty thanks to Ms. Danya for anchoring this event nicely with dedication. And finally, I express my sincere thanks to all my dear colleagues whose handholding, support, and unity has made this event a grand success. Once again, thank you all. Stay safe, healthy. Have a nice day.
Thank you, Sujata Miss. The link for the feedback form for the session will be posted in the chat box. Kindly fill the form for receiving your e-certificate. The link will also be posted in the WhatsApp group. For those who were not able to join the live session, we'll be providing you the link for the meeting recording and feedback form later. Once again, thank you all for your cooperation. We can now exit the meet.
Thank you.
Thank you, sir.